Hello future doctors, how are you all? Welcome back to NEET Steps YouTube channel. So before going to video, I'm going to ask one question. You know which bacteria can't be seen on a gram stain, or which rod shaped bugs cause deadly toxins? So friends, today let's break down bacterial taxonomy and morphology. So if you're understanding these things, then you can easily find out which bacteria can be seen on a gram stain and or which rod shaped bugs cause deadly toxins. So before going to video, let me introduce myself. I'm Anand Kumar Jana, final year medical student. So without time waste, let's start. So today our topic is bacterial taxonomy in microbiology from first aid guide. So bacterial taxonomy means the naming and classifying microorganisms. The bacteria are classified based on shape we called morphology, and cellular structure. With gram staining we can divide it into gram positive and gram negative, and based on oxygen requirements. And special staining we have with like gram stain, acidic fast, silver stain and Jimsa. These are the special stains. And we have some growth characteristics and genetic traits like plasmids and DNA. So individually we are going to discuss in gram positive and gram negative based on morphological structure. So first one, morphology means shape and gram stain. So first of all we are going to see the spherical shape, means coccus. In coccus, for example, we can see staphylococcus and streptococcus. So in gram negative we can see like Moraxella, Catarrhalis and Neisseria. So in rod morphology we can see Bacillus. In Bacillus the gram positive examples will be like Bacillus, Clostridium, Corynebacterium, Gardnerella and Lactobacillus, Listeria, Mycobacterium and Propionibacterium. These are the examples of gram positive bacteria in rods. Next one, in gram negative we can see based on system. First one, enteric, means gastroenteric causing pathogens or microorganisms like bacterioids, Campylobacter, E. coli, Enterobacter, Helicobacter, Klebsiella, Proteus, Pseudomonas, Salmonella, Serratia, Shigella, Vibrio, Yersinia. In respiratory system we can see like Bordetella, Haemophilus and Legionella. In zoonotic we can see Bottonella, Brucella, Francisella and Pastorella. Okay, so in branching filaments mostly we can see in gram positive Actinomyces and Nocardia, and in negative we can't see anything. So next one we can see pleomorphic. Pleomorphic means it's like variable shapes. For example, Haemophilus and Mycoplasma, and you can see like Chlamydia and Rickettsia. And we have some spiral also. In spiral we can see spirochetes like Borrelia and Leptospira and Treponema, and we have some no cell wall morphological things like Mycoplasma, Ureaplasma. Okay, and these things are very resistant to beta lactams because it contains sterols which don't gram stain. Okay, so here I am going to divide one thing, that is gram stain. This is by gram stain we can divide into two parts. First one gram positive, next one gram negative. So in gram positive we can see that stain will be like purple. In gram negative we can see the stain will be pink in color. Okay, so in gram positive the cocci, in gram positive the purple stain we can see in cocci, Staphylococcus and Streptococcus. In gram negative we can see in cocci, Neisseria and Moraxella. Next we can see in rods like Bacillus, Clostridium and we can see Listeria, Corynebacterium. So these are the rods. So in gram negative we can see rods will be like in enteric, E. coli, Klebsiella, Salmonella, Shigella, Vibrio and Yersinia. Okay, in respiratory system we can see Haemophilus, Legionella and Bordetella. In zoonotic bugs we can see like Bartonella, Brucella and Francisella and Pastorella. And we have some special strains, that is Giemsa strain. The Giemsa strain will be like Chlamydia and Rickettsia and Borrelia we can see. And in silver stain we can see like Legionella, and we can see like acid fast stain like Mycobacterium and Nocardia. Okay, next our topic will be microbial pathogenicity. The microbial pathogenicity means we have to focus on how bacteria cause disease. This thing we call microbial pathogenicity. A bacterium becomes pathogenic based on its virulence factor or virulence properties. So we can see here these are the genes. These are the genes products from my chromosomes or plasmids. First of all, we are going to discuss about virulence factors. In virulence factors, based on the chromosomal and extra chromosomal like plasmids, gene products will related to the organisms. In virulence factor, first of all, we have to focus on invasion properties. In invasion properties means how that the entry into the host cells or tissues will becomes in invasion properties. Next one, adherence and colonization. In adherence and colonization we can see binding via pili and adhesions, and we can see some toxins. The toxins in toxins we can see like the tissue damage induced toxins like exotoxin and endotoxin. In exotoxin will be like it's produced and secreted, example botulinum toxin, and we can see endotoxin is like part of cell wall like lipopolysaccharides in gram negative. Next one, immune system reactions, or you can call immune evasion. In immune system reactions we can see capsule, example like streptococcus and pneumonia, antigenic variation like example Neisseria and gonorrhea, and we can see biofilms, example like Pseudomonas. And we can see some like intracellular growth, the intracellular growth or intrasurvival, example like Listeria and Mycobacterium. And like eluding host defense mechanisms and antibiotic resistance. In antibiotic resistance via like plasmids, efflux pumps, enzymes, examples like beta lactamase. So a normal flora may become pathogenic if they gain access to normally sterile body areas, or the environmental conditions allow them to multiply to a level not controlled by the host. So here we are going to see the microbes pathogenicity to related with colonization. Colonization is the main important thing because it is the first step of infection. The colonization it established a foothold at the entry site. Like common entry points we have, that is respiratory tract and gastrointestinal tract, urogenital tract and conjunctiva. In conjunctiva we can see like eyes. These areas are lined with mucus and often exposed to the external environment. If the immune system fails to respond effectively at this stage, that time infection will begins. Okay, so we already discussed this thing and this thing. Yeah, this thing also we discussed. So in clinically, what are the things important? We can see when infection is begins, that time we can see like some symptoms are portal of entry, like organ systems involved, like risk factors like that risk factors will be like immunocompromised host or patient. If for investigation we can do gram stain or culture, and special stains like acid fast, silver and Giemsa, and polymerase chain reaction, PCR rapid detection and antibiotic susceptibility tests. So we can manage with some antibiotics, that is like empirical antibiotics based on gram status. In gram positive mostly we can use, okay, in antibiotics, if the patient have gram positive organisms, then we can use penicillin and vancomycin. If that patient have gram negative, then we can use cephalosporins, okay, and fluoroquinolones and carbapenems. And in atypical we can say like macrolides and tetracyclines also we can use. And we can tailor the antibiotics based on culture or sensitivity, and we have to control the infection with the hand hygiene, isolated when needed, and sterile procedures. And we can prevent with prophylaxis in exposed patients and vaccines like Haemophilus influenza and pneumonia, streptopneumonia and Neisseria meningitidis. Okay, so these are the things we have to follow, and this is the management. So we already discussed about colonization. In colonization, most host defense mechanism at the surfaces like mucus, cilia, lysozymes, IgE antibodies, normal flora competition. To overcome these pathogens need adherence mechanisms. So now we are going to discuss about adherence mechanisms. Means adherence to cell surface involves, here we are going to discuss about adherence. This adherence mechanism means how the bacteria stick to surfaces. How did the bacteria stick to surfaces? This is the adhesions. The surface protein or toxins that bind bacteria to specific receptors, like example pertussis toxin and heme-agglutinins, like it bind to respiratory epithelium. So next one we can see the pili and fimbriae. Here pili and fimbriae means it's like hair-like protein structures in gram-negative bacteria. Actually, before this video I made one video about bacterial structures in microbiology from first aid guide. So if you didn't watch that video, go and watch that video. Then after you will get this video, you will get like little deep knowledge about the bacterial structures. Okay, so because I already covered every basic about bacterial structure, so if you are seeing that video you will get a good idea. Okay, so this pili fimbriae will be like hair-like protein structures in gram-negative bacteria, and it help with specific binding to host cells receptors. Example like E. coli, Neisseria and gonorrhea. Okay, next one we are going to see the lipoteichoic acids. Lipoteichoic acids means it's mostly found in gram-positive bacteria cell wall component. So its role in non-specific adherents. Example like Streptococcus pyogenes. Then after we can see IgA proteases. IgA proteases means IgA it's like enzymes that cleave IgA antibodies, and we have some Fc region on antibody like of IgA, then sticks to mucosal cells and helps bacterial attachment. Examples like syn organisms. Syn means mnemonic, like Streptopneumoniae and Haemophilus influenza type B and Neisseria saprophyticus. Okay, so next one we can see here biofilm. Biofilm means the bacterial fortress. Biofilm means it's like biofilm means it's a protective community of bacteria inside an extracellular polysaccharide matrix, also known as biofilm. So first of all the bacteria will attach, then after we can see the bacteria will multiply. First the bacteria will attach, then after it will multiply, then after it secretes matrix, and it build biofilm, then after it resist immune system and antibiotics. Okay, this is very important, that's why I am going little deep. so we will discuss another time. First one, the bacteria will attach to host cell, then after it will multiply, then after it secretes matrix, then after it build biofilm, then after we can see it's resist immune system and antibiotics. So what are the steps we can see in biofilms? First of all, the steps of biofilm formation will be, first of all, it will get attachment to a surface, example like catheter or prosthetics. Then after it will form some coating with host proteins, example like fibrinogen and fibronectin, and then after we can see maturation. In maturation it will forms like into a protective layer, and then after we can see dispersal of individual cells like planktonic bacteria, and it spreads infection. Okay, then after it will spreads the infection. This planktonic cells is very important. Okay, so first thing is attachment of a surface to a surface, then after coating with host proteins, and we can see maturation into a protective layer, and we can see dispersal of individual cells. Why these biofilms are dangerous? Because it block antibiotics from entering, and it resist opsonization neutrophils and T cells, and it cause chronic infections. And main thing will be like it require removal of foreign bodies for definitive treatment. Okay, so now we are going to see the one small table, that is in vivo biofilm forming bacteria. The bacteria will be like first of all, the bacteria is S. epidermidis. The streptoepidermidis will be like the clinical conditions we can say like catheter infections, prosthetic valves and shunt. And we can see next one, Virulence, streptococci mutans and sanguinis. The clinical condition will be like it will do like dental plaques and infective endocarditis. Next one, Pseudomonas aeruginosa. In Pseudomonas aeruginosa, the clinical conditions we can see like cystic fibrosis, lung infections, ventilator pneumonia, and in non-typable hemophilus influenza we can see like clinical conditions like otitis media, conjunctivitis and sinusitis. So the clinical importance things will be, if you suspect about biofilm related infections like chronic or recurrent infections despite antibiotics, and presence of foreign bodies like catheters, pacemakers and prosthetics, and we can see like cultures negative but infection signs persist, and we can do some test, that is blood culture, device tip culture and imaging like ultrasound, CT for abscess, and we can do some molecular tests like PCR and 16 sub S rRNA. Okay, how we can do management? So for this biofilm we can do management. How we can do management? First of all, we have to remove the device. What are the devices you keep in that patient? We have to remove the things. Okay, if possible. Like this is like definitive in biofilm infections. Next one, we have to start high dose or combination antibiotics. High dose of or combination antibiotics means example like rifampin plus vancomycin for prosthetic joint infections. Okay, next we can do like prolonged treatment, like often required like 4 to 6 weeks intravenous prolonged treatment. And we can start like target biofilm resistant strains with like daptomycin, linezolid and fluoroquinolones, example like ciprofloxacin for Pseudomonas. Okay, so these are the things we have to focus. Next we are going to see the like bacterial immune evasion mechanisms. Okay, so here main thing is Now we are going to see the how we can avoiding immediate destruction by host defense systems. So in basics we have to focus on antiphagocytic surface components which inhibits phagocytic uptake. Here some bacteria wear an invisibility cloak. Let's look at two examples. First one, capsule. The capsules we can see like polysaccharides layers that hide the bacteria from immune detection. Examples like Streptococcus pyogenes uses a hyaluronic acid capsule that mimics human connective tissue, helping it to evade phagocytosis. In clinically, this capsule will be like encapsulated organisms are poorly immunogenic, meaning vaccines often need conjugation with a carrier proteins. How we can do diagnosis? We have to use Quilling reaction to visualize capsule swelling. So for management we have to do like capsule targeted vaccines like pneumococcal vaccine PCV13, PPSV23 and Haemophilus and we can give some HIV vaccines and meningococcal vaccines. Next one, IgA proteases are enzymes that cleaves IgA. We already discussed this thing. It's secreted by syn organisms like S. pneumonia and Haemophilus influenza type B and Neisseria streptopyticus. IgA protects mucosal surfaces, so this bacteria cleave it, making colonization of the respiratory mucosa easier. Okay, so the clinical relevance are commonly involved like otitis media, okay, sinusitis and meningitis, pneumonia. So how we can do management means we can use vaccination and prompt antibiotic therapy for those pathogens like amoxicillin, clavulanate, or we can tell augmentin or ceftriaxone for severe infections. Next one, antigenic variation. Antigenic variations means ever wonder why you get recurrent infections from the same bug? Let's blame antigenic variation. So in definition we can see like microbes changes surfaces antigens to evade memory immunity and reinfect the host. For example, we can say Neisseria gonorrhea and it changes pili and outer membrane proteins, Enterobacteriaceae like varies capsular and flagellar antigens. The clinical challenge will be no long-term immunity and repeated infections. Vaccine development is difficult for highly variable organisms like HIV, malaria and gonorrhea. How we can do diagnosis means we can do polymerase chain reaction, PCR or antigen specific detection based on current variant. Okay. So ability to survive intracellularly. The intracellularly survival means it hiding inside host cells. Some bacteria hide inside the host cells, escaping like immune detection. That mechanism will be like, first of all, the Mycobacterium tuberculosis. In Mycobacterium tuberculosis, we can see it survives by inhibiting the phagosome and lysosomal fusion. It survives with macrophages. Next, we can see Listeria. This Listeria quickly escapes the phagosome into cytoplasm before the phagosome lysosome fusion. So this Listeria monocytogenes, it escapes phagosome to replicate in the cytoplasm. For diagnosis, we have to do AFP stain for TB, and we have to do cerebrospinal fluid or blood culture for Listeria in meningitis. For management, we can do for TB long-term multi-drug therapy like RIPE regimen, and Listeria like intravenous ampicillin with or without gentamicin, especially in neonates and immunocompromised. So next one, type 3 secretion systems. In type 3 secretion systems, we can see like this is like biological warfare. Bacteria directly inject toxins into host cells. Example like Pseudomonas, Salmonella, Shigella and E. coli. This bacteria use a needle-like structure to deliver virulence factors into host cells. Example like watery or bloody diarrhea, tissue necrosis or inflammation, and pneumonia or pseudomonas. How we can diagnose? We have to do culture with virulence gene analysis, PCR or toxin assay. For management, we have to do supportive care and targeted antibiotics. In Shigella, we have to avoid anti-motility drugs. Next one, inflammation or immune mediated damage. The immune mediated damage and inflammation will be like that sometimes our own immune system causes the problem. For example, rheumatic fever. In rheumatic fever, we can see like cross reactivity of anti-streptococcal antibodies with heart tissues. We can prevent with prompt penicillin treatment instead of pharyngitis. So next we can do tuberculosis. Next we can see like tuberculosis and leprosy. In tuberculosis, leprosy, we can see the granulomas and tissue destruction. For treatment, we have to do long-term antimicrobial therapy. Next one, post-streptococcal glomerulonephritis. In post-streptococcal glomerulonephritis, we can see like the immune complex deposition in kidneys. For management, we have to do supportive care and antihypertensive drugs and diuretics. The structural toxins will be like gram-positive fragments like peptidoglycan and tichoic acid. It stimulates inflammation even without active infection. Next, we can see physical damage or pathogen size or location. For example, in physical damage, mostly we can see some swellings from infection in a fixed space damaged tissues, and we can see like large physical size of organisms may cause problems. Here, for example, the size matters are where the pathogen sets up shop. Example like Ascaris lumbricoides. The Ascaris lumbricoides intestinal or biliary obstruction can cause Entamoeba histolytica. It can cause ulcerates colon, can lead to liver abscess. And we can see like cysticercosis, like brain cysts causing seizures. And we can see like bacterial meningitis. It can cause swelling damages brain tissue due to fixed cranial space. For management, we can do like antihelminthics, albendazole for Ascaris, and metronidazole for Entamoeba histolytica, and corticosteroids for central nervous system inflammation. Example like TB meningitis, tuberculosis meningitis. And we can do surgical intervention if obstruction or mass effect occurs. Okay. So in summary, every microbes have evolved multiple tricks like hiding with capsules, cutting antibodies with enzymes, changing their surfaces like a costume party, and injecting toxins, living inside our cells, or letting our own immune system do the dirty work. So we can have some easy mnemonic for USMLE, that is change tip. Change tip means changi tip means CHANGI TIP tip. Changi means C. First C means capsule, H means hiding intracellularly, A means antigenic variation, N means needle-like secretion, G means granuloma like immune damage, IgA protease, T means toxins, I means immunocomplex, P means physical blockage. So always consider these mechanisms when you face recurrent like chronic or immune complicated infections. Okay. So mostly we discussed about bacterial taxonomy and some main things of bacterial structures, and we studied some antigenic variations, how that colonization will happen, everything we discussed, and how our immunity will affect against and us, everything we studied in this video. If you have any doubts about this about any topic, you can drop one message in comment section. Based on that, I will give reply. If this video is helpful for you, like, share and subscribe. We will meet in our next video. Our next topic will be like gram stain in microbiology from first aid grade. Okay, so we will meet in our next video. Till that time, take care. Bye bye.